I'm so excited to be here in Barcelona in front of the real people. So, Hanna Lesch, Chief Technology Officer from Exothera, and today I'm going to present shortly who is Exothera, and a little bit I will talk also on the things you should consider during your product lifecycle, and also how Exothera is responding to those. But first of all, who we are. So Exothera is a subsidiary of Universals. So Universals is founded 2013 by Jose Castillo and Hugo Pultod, and Jose is an inventor of the fixed bed bioreactors. And they had a vision to provide biologics for all. Key affiliate for our field is Universals Technology, who provides manufacturing solutions for manufacturing for biologics. So Exothera, it's a contract and development organization focusing on viral vectors and vaccine manufacturing. So we are a full-service CDMO. So what does it mean? It means that we want to be a one-stop shop for our clients, so that when they come to us, they don't need to go or anywhere else. So we are serving our clients from the beginning to the end. We can provide plasmid supply, but it means that we don't have a plasmid manufacturing in-house today, at least, but we can provide plasmids for our clients through our selected business partners. It's also the same if client needs help with the vector development, we have a broad networks. We can do cell banking, process development, analytical development, manufacturing, fill and finish soon, and of course always supported by regulatory. Here is Exothera through some figures. I will start from the middle because there is people. So today there is 140 of us, I guess even maybe 150. We have two GMP facilities, total of 2,100 square meters. It's brand new, and we just recently got an approval from authorities for clinical manufacturing. Funding is well secured, and some numbers in the scales that we can work, 2,000 liters stirred tanks and up to 600 square meter culture area for adherent cells. And today we have conducted, or we are doing 28 projects so far. So we are located in Belgium. In Zumé, we have our main offices, our main facilities. So in Zumé, we have a Vega building and Nova building. So Vega is our GMP facility for large-scale manufacturing, and in Nova building, we have a small-scale GMP facility, and then we have our offices and development laboratories, QC, in Nova building, as well as the main warehouse. Then we have also our old facility still that we're using. It's in Gosselies for R&D purposes. It's just 5 minutes driving away from Zumé. And here you can see a fancy picture of our facilities, and you can see that the buildings are really, really close to each other. So Exothera is 2 years old only, but we already have a nice proven track record of the work we have done. So 28 projects, and I have to say that I have been in a company for 6 months, and this was something was impressing me, and this has been only possible because we have very experienced team members working. Publicly disclosed collaborations, you can see there. We have been working with lentiviral adenos AAVs and with several vaccines and different cell lines. Go. So that was Exothera in a nutshell, and then I'm a little bit going through also the points that you should really consider during your product life cycle, and then how we can respond to those. But everything starts from the construct design. So if you have a rusty parts, it will be very difficult for you to build a shiny car. And it's the same with the viral vectors. So basically everything starts from your construct design. Pay attention on your transgene and the expression cassette you have. And really critical is also your vector backbone that you are using in your trials. It's known today that you can improve the quality and productivity by construct design. And there are several groups, for example, who have been improving the AAV vectors and then have been able to show a better empty versus full ratio, for example. If you're using plasmids, should you use already today antibiotic-free systems? Maybe not necessary, but one thing to consider, at least for the future, but at least kanamycin should be used from the beginning. And then I come from the research and development background, so one thing I can also advise you to do is sequence your construct as early as possible, just to avoid any unpleasant surprises on your way. Then when the product is going to preclinical efficacy studies, and then of course to regulatory required tox and safety studies, you should already pay attention on how do you do your virus for these preclinical studies. So you should fix your construct, use the same construct through the whole experiments, manufacture it with the process that is comparable that you're going to use in later with the material, raw material that are not maybe in early R&D stage GMP compliant, but that you can find those materials then later in GMP compliant manner and use the same cell line for manufacturing. And better to use adequate analytical methods and really focus on that. More is better than less. Then the next thing you need to consider is actually a critical decision on your manufacturing strategy. So are you working with lentivials or AAVs? Quite often people are still using plasmid transfection. It's fine, it's working, it's very fast, it's flexible. Easy to kind of today go to clinical trials, but then you will face definitely later on challenges with large-scale manufacturing, and especially then it's very expensive to make a plasmid transfection in large scale. If you're working with, for example, adenovirus, you can use infection, then it's a little bit more straightforward and more cost-effective manufacturing. Today, there's still reasonably small amount of stable cell lines used and even commercially available, but it's one thing really heavily to consider that if you can use stable cell lines, then your manufacturing later on will be more cost-effective, straightforward, and definitely a lack of even certain challenging steps. But stable cell line development can take time and can be very expensive at the beginning. So then we come to the manufacturing process and technologies around that. So here, as I mentioned, is a bioreactor, fixed-bed bioreactor from Universal Technologies, the Scalax, and Exothera has been one pioneer working with these bioreactor types. So Scalax family, it's a fixed-bed, disposable, scalable bioreactor providing homogeneous three-dimensional layer for cells to attach. Scalax Hydro, it's the smaller scale, 2.4 square meter culture area, and excellent tool to make a virus, for example, for preclinical studies or use this in process development. Scalax Carbo, it's the middle scale bioreactor, and then Scalax Nitro, you can have 200 or even 600 square meter culture area in very compact footprint. And Scalax technology has proven to be very applicable for different processes and different viruses. So it has been used, for example, for AAV manufacturing using plasmid transfection. And the first graph here is a proof when showing very straightforward transfer from the cell culture flask directly to the bioreactor. Then the next graph is proving the lentivirus production in this system with plasmid transfection system, and the technology is really providing already easy perfusion, and in this study, for example, the feeding strategy was optimized, and even through that the productivity was improved. It has been also proven to be efficient for adenovirus production, for example, and in this last case using the serum-free media. So fixed-bed bioreactor is an excellent tool to provide a scalable method for adherent cells, but it doesn't mean that it can be only used for adherent cells. It has also gained a lot of interest nowadays to use it in suspension cells. So you can use, like we have done here, the work with suspension cells to produce adenovirus. It means that you use a suspension cells, suspension conditions, and then the cells are actually entrapped by the fixed bed. They are not attached there. The cells don't have those arms in the suspension condition really to hold tight on that, but they are attached and entrapped into the fixed bed, which is then providing a very high cell density in a small compact volume. In this study, we use suspension cells and optimize the cell density for adenovirus production. And basically what we can see from this graph is that, well, if more you have cells and happy cells, more they produce virus. Exothera is not only providing ScaleX technology. We want to meet our clients' needs, whatever they are. And if client is interested to use suspension cells and stirred tanks, we are relying on Pulse Allegro system. So we have 200-liter and 2,000-liter stirred tanks in-house. And if we are doing process development, we are using the DASKIP and DOE, design of experiment, to optimize the critical process parameters in a small scale before scaling up. And these stirred tanks are also shown to be very applicable for different viruses and different kind of processes. The first case study shows the AAV production using stable cell line, using the CEVEX Elevector cells from 10-liter to 200-liter scale. They were able to keep the same productivity. The second case study is showing AAV manufacturing using PEI-based plasmid transfection in 50-liter and 200-liter scale. And the last case example is using Allegro system in 1,000-liter suspension for adenovirus production. And not only that you need to have an upstream solutions, we are relying on very standard, disposable and scalable downstream using the filtration and chromatographic purification. And one thing we can provide also for our clients, it's a methodology to support our client in these critical decisions. It means that a client can come with their current process to us, and we use our expertise and our professional team to really evaluate the current process and then make proposals. It may or may not contain some laboratory work, and this way we can support our clients in their decision making. It doesn't mean that the client needs to continue with us, but of course that's something we hope, but it's not necessary. And always key is to have regulatory support from the very beginning. Why? Is because you want to do first time right your product development to save time and money. And why is that? Because at the end we are aiming to have authority approval for this product, maybe to clinical trials or later even on marketing authorization. So pay attention that your regulatory story is solid through the way. So basically, I went through quite fast many critical things that you need to take into account. And then I will conclude my presentation just saying that find the right partner who can support your development and manufacturing process. So thank you very much. If you are interested on Exothera or our services, you can contact me, or I'm here with my colleague from Nivelles, who is our Vice President of Business. And then there's also, if you're interested in ScaleX technology, there's Universes also presenting in this conference, like, for example, Maria Jordan is there in the audience. Thank you very much.